Hello students, so today in this video we will be studying about microsporogenesis. Students, microsporogenesis is the process of formation of microspores. Microspores are the spores that give rise to male gametophyte, which is the pollen grain. The male gametophyte consists of the male gamete. That is, the pollen grain will contain the male gamete. Now students, we know that this is a diagram of a transverse sectioned anther. Here we have the anther wall. The anther consists of four sac-like structures called microsporangium. These microsporangium are filled with cells. These cells are known as sporogenous cells. They are compactly arranged inside the microsporangium. Now students, when the anther is young, the microsporangiums are compactly filled with the cells. But as the anther grows or as the anther matures, the size of the cells also increases. And as the size of the cells inside the microsporangium increases, the size of the microsporangium also increases, as a result of which the cells which was previously compactly packed now develop space in between them. And now after the anther matures, we start calling the sac as pollen sac. So students, when the anther is young, the sac is known as microsporangium. But when the anther matures, the sac is known as pollen sac. Now, all these cells inside the pollen sac are capable of producing microspores. But only few of them give rise to microspores. Rest of them gets degenerated. The cells which gives rise to microspores are known as microspore mother cells. Now students, let us see how these microspore mother cells give rise to microspores. That is the process of microsporogenesis. These microspore mother cells are diploid cells, which means they have 46 number of chromosomes. These cells will first undergo one meiotic division, that is meiosis, as a result of which we will get microspore tetrad, that is 4 microspores will be produced which are attached to each other. These 4 microspores will be haploid. Why haploid, students? Because the microspore mother cell underwent meiotic cell division, which is a reductional cell division. It reduces the chromosome number into half. Now, these microspore tetrads in the next step will get separated from each other, and these individual cells are known as microspores. So students, this was microsporogenesis. We saw how the microsporangium cells gives rise to microspores. Now we will see how these microspores gives rise to the male gametophyte, which is the pollen grain. So students, these microspores will now form two layers around them. The first inner layer is known as the intine. Surrounding the intine is the second layer, which is known as the exine. The exine has irregular surfaces. This exine was released by the fourth layer of the anther wall tapetum. This layer exine is made up of sporopollenin. The gaps in between the exine are known as germ pores. So students, this structure of microspore is known as the pollen grain. So pollen grain is actually a microspore with the layers intine and exine around it. So students, this is how a microspore gives rise to a pollen grain. Now, this pollen grain will undergo one mitotic cell division. So, the cell first will undergo karyokinesis, that is nucleus division. So, the nucleus inside the pollen grain will get divided into two. Then the cell will undergo cytokinesis. Now students, the cytokinesis is not equal. After cytokinesis, we get one larger cell and one smaller cell. The larger cell is known as the vegetative cell, which do not take part in reproduction, and the smaller cell is known as the generative cell. This cell is the one that will give rise to the pollen tube. Now the pollen grain, which is at 2-cell stage, is now ready to get released from the anther. So during this time, this parts of the anther gets rigid, except these middle portions of the cell. These middle portions ruptures and the pollen grains gets released from inside the anther. This is known as dehiscence of the anther cell.